Hello, everyone. Happy Easter. Welcome to another coffee chat session of my YouTube channel based on data and science. In today's session, we'll actually discuss about the implementation of AI or machine learning in scientific research. I'm honored to join by Lukas, an esteemed guest from academia, who will talk about his experience about how he's implementing AI machine learning or data science in his day-to-day academic research work. Welcome, Lukas. Thanks for joining our coffee chat session today.
Hi, Sam. Thanks for having me.
So with further ado, I'm going to actually ask Lukas, if you just introduce yourself to my viewers before we dive into the main section of this coffee chat session.
Sure. So I'm Lukas Simon, and I'm a group leader of computational biology at the Therapeutic Innovation Center at Baylor College of Medicine in Houston, Texas. And I lead a small group of data scientists with the goal to apply state-of-the-art data science to answer questions on the level of molecular data, specifically with focus on developing early cancer therapeutics.
That's very fascinating to know, and thanks, Lukas, for providing the kind of like a groundbreaking research work your team has been doing. That will have a lot of implementation in future, and hopefully it will change the paradigm of artificial intelligence or machine learning in the scientific research world. So with further ado, I'm going to dive into the main section for today's coffee chat session. So I'm going to start with the first question to you, Lukas, is, in your opinion, what's the significance of data in your scientific research?
Yeah, I think that biology or biomedicine has evolved in the last 20 years or so, just like many other fields have, in the way that technology has advanced to a point where it's very easy to collect very large amounts of data in your routine experiments. Specifically, I'm referring to one of my topics of interest, which is gene expression. So technology has advanced in such a way, with the advent of what's called next generation sequencing, you're able to measure all genes in your sample with relative ease. So if you think about the data that it produces, we can think about kind of ballpark numbers is that you have approximately 20,000 genes that are expressed in any cell. And so if you profile a couple of samples, you will end up with a data matrix that captures 20,000 genes across the number of samples that you have. Mm-hmm. And so that's for example, just a relatively large matrix that is, at the one hand, it's challenging because it's not easy you can't just look at it and know exactly which gene is sort of important here. At the same time, it's super exciting because data analysts can apply sort of more sophisticated analysis methods in order to ask questions or try to answer questions.
No, that's definitely fascinating to know about that, because as you mentioned about the collecting the gene expression data, like we know that after the advent of Illumina, and how quickly we can literally produce thousands of millions of gene expression data, and we all know that it's kind of difficult to learn a pattern from a gene data, so that's why we kind of put it in that unsupervised learning category. But it's fascinating to know that, and every day your research is evolved around the gene expression data. Just try to understand the pattern, and that's I believe it's one of the, I guess, big segue to understand that how we can manipulate or process the gene data. Maybe that will give us some sort of a hidden meaning of what's going on. So yeah, thanks for providing that information of what kind of how you're actually using data for your scientific research. We're talking about millions of records processed like a few every day. So yeah, so that's kind of literally take me to kind of like the next question, because that's also related to like a data, because as you mentioned that your team is being very involved in the processing the gene data and biological using like different biological contexts and applying AI and machine learning or data analysis work. So as a data scientist, I always try to always feel like that most of the time we spend more time in processing the data rather than building a model based on that, right? So most of the time we perform data wrangling or some sort of data cleaning to understand that we have a clean or tidy data set that can be fit into a model. So based on your daily experience or some day-to-day work, so how challenging is to like collect the data, like source the data, manage the data, and also transform it into a kind of a relevant form so that you can build a model, or at least you can get understand some sort of underlying pattern from the data, especially in terms of gene expression data or any other molecular biology related data.
Yeah, that's a very interesting question, and I have sort of my own kind of personal definition between two subfields of computational biology or bioinformatics. And I don't know if everybody will agree, but for my own, I mark a certain boundary in the analysis. So what I call bioinformatics or computational biology, sometimes people use these two terms like interchangeably, but I make my own kind of definition. And I say bioinformatics is everything that happens before you get your sort of data matrix. So for example, if you're talking about gene expression, there would be a gene expression matrix. So what algorithm, what steps are involved in order to create that matrix? And then computational biology is what happens downstream. So in computational biology, you take that data matrix and you do sort of analysis, maybe PCA or some other models, in order to gain insight from that matrix. That's also sort of my own personal definition, where I can put a clear boundary between these two topics.
Yeah.
And I think so it really depends on sort of the research focus that people have in these standard processing pipelines. So ways to get from your raw data, which is actually sort of reads, how to get to account metrics. There are established pipelines, and some research groups, they focus on how to improve the pipeline and maybe how to gain more insight from different aspects of this pipeline. But a lot of researchers just kind of live and accept the standard or the state-of-the-art pipeline. They just run that, and then they focus their research on the downstream part, how to maybe model the count metrics and so on. So I think there are challenges in both ways. So maybe let's talk about a little bit computational sort of I would say hardware challenges. The data during the bioinformatics step is very large. So you're talking about a single read in any given sample. You have approximately 50 million reads. Then for each read, you also sort of measure some sort of quality scores and so on. So just for a single sample, right, you would have a text matrix or a data file that would have approximately 200 million rows. So it can be a very large file. And now that's just a single sample. Let's say you have a study where you're comparing healthy and maybe diseased patients, then that can be hundreds of samples. And so now you're talking about gigabytes to terabytes of data. And of course, depending on what the project is, this can be very big. So during that step, that bioinformatics step of saving the raw data, processing the raw data, getting it to a data matrix analysis kind of format, there's a lot of challenge, just very sort of IT related challenges, right? Such as storage, such as running algorithms in a relatively fast time to process the data, and so on. So there are these, I would call them kind of, hardware challenges that are things that need to be considered, that maybe at the computational biology perspective, so downstream analysis, you don't really have to think about that much. But I think also there's a lot of opportunity for discovery when you think about, okay, this is the state-of-the-art algorithm. We all kind of know that you can do these following steps to quantify gene expression. Is there an opportunity to learn something more, learn something new about it? And for example, in our team, we have also taken the standard analysis, so just counting reads and trying to quantify gene expression. And we thought, okay, can we be a little bit smarter about it? And can we quantify different parts of gene expression in different ways? So we developed an algorithm that allowed us to quantify specific aspects of gene regulation in a sort of non-traditional way. And so now looking back at the big picture, I think one of the questions you asked, okay, where can you use AI in this whole long step? I do think that there's this concept of feature engineering, right? Or trying to kind of take the raw data
feature from the raw data. Yeah.
Right. Have maybe a more creative or innovative way to summarize your raw data, and that maybe allows you to come up with new discoveries as well. So I think there's a whole spectrum of analysis, and there's also a whole spectrum of skills that people have to have in order to kind of work along this pipeline. But I would say that as you go further away from the raw data, you're probably dealing with less data and less sort of maybe hardware challenges, and more towards the modeling. So people that really like modeling, I think sometimes I feel like they prefer to work on a nice count metrics, and they don't really care how the count metrics came about, but it's going to be nice to be handled.
Yeah. No, I think that's really true, because in most of the cases, people just think about an outcome of the model rather than how much time it gets spent to develop the data to make it compatible with the model. So we usually less tend to focus on developing the data structure. So just out of curiosity, so in your academic research, do you usually partner with in a group of data engineers or IT team who can help you out in processing the data and also building the metrics that you can use for your model building exercise? Or usually your team are kind of responsible to perform the data processing step?
Yeah, I think maybe there's not a single rule, but I think the most common approach is that each, let's say, computational lab or computational group, they will have maybe one person who's going to be more familiar with, kind of IT or admin, and kind of, let's say they're very comfortable living in like a high performance computing environment, and know how to install programs and run programs and submit it to a queue and so on. But of course, every department generally provides the hardware, so you can kind of have access to a high performance computing system, and they will provide also some IT support. But generally, things such as running the pipeline, maybe installing the algorithm and so on, that's something that's usually handled within the team. The IT support is sort of more high level. If you were to lose some storage, maybe they can try to recover it, or they can make sure that all the computers are running the way they should be.
No, that's great. Thanks, Lukas, for providing this additional information, because it's kind of like a very interesting segue. I mean, a very interesting setup that you have, where you do already have your data engineers or somebody with an IT expertise within your lab, so you don't actually have to spend an additional time to partner with maybe third party or somebody else. So I feel like it's more of like a cohesive approach to have all the kind of experts within your lab. So you won't spend a lot of time to processing the data or getting the data from outsource. areas. So yeah, that's really great to know. I'm pretty sure some of my viewers in the industry area, they would actually kind of relate this to the this experience, because in the industry area we do also have kind of a team, like a data engineer or data analyst or data scientist. But most of the cases they tend to like work very cohesively. I mean, their work has been kind of like overlap with each other whenever they run any project, or whenever they're involved in processing any data through a pipeline. And so I could literally relate to that with what you just described about your data processing pipeline involving data engineers or somebody from IT department. So thanks, Lucas, for that. So now that's kind of take me to kind of like the last question for this coffee chat session, and that's more of this question is more of like a futuristic, because as you mentioned that like when we process the data, we formulate into matrix format, and then after that maybe that feature engineering thing come up. And in recent days we have seen that the advent of like the deep learning, that how deep learning is being very useful in terms of taking the paradigm of AI to the next level, and also it helping the data scientists or AI developers to extract feature automatically, because that's kind of like one of the limitation of machine learning, where you basically have to involve to extract the feature, you have to compute that, you have to code that. But in deep learning, it usually kind of like derive the feature engineering part automatically by using the neural network. So having said that, given this kind of technology advancement in the deep learning and machine learning area, so what do you think, how would your scientific research or the kind of work you do would get benefited from the advancement in the deep learning space? Do you think like your team will be more go towards deep learning space in order to build a model, or do you think it will be more kind of still related to just a machine learning or the basic level of machine learning technology that your team would use in future as well?
Yeah, that's a interesting question, and I think you're absolutely right that in recent years we've seen more applications of deep learning in biology or biomedicine than before. And actually, just to give a little bit more context, I kind of want to maybe introduce, I think, two major changes or steps that have, I think, really led to the application of deep learning in our current research. So one is, I think I mentioned it kind of before, is that in a single experiment you can measure thousands, 10,000s of genes, right? And so when we talk about application of deep learning as sort of as a rule of thumb, is that you want to have around 10,000 data points or so in order to apply your model. So now it really depends on what is the question that you're asking and what are you using as data points. And so let's think about a typical gene expression matrix. It will have maybe the genes as rows. So you have like 20,000 rows, and then maybe you have the number of data points as columns. And so there was a specific advancement in the technology, which is called single cell RNA sequencing analysis, which allowed us to profile gene expression, not just across previously it was called bulk RNA sequencing. So there you look at like thousands of cells and you just kind of average the gene expression across those cells. But now we have the ability to actually sequence a single cell and measure the expression levels in a single cell. And this technology has nicely matured in such a way that in a regular experiment, you can profile between 10,000 to maybe 1 million cells. And now I just want to kind of, again, put that back into context where you think about the data matrix. Now you have 20,000 genes and now you have 1 million columns. So that's actually a huge matrix. And it really changed the whole realm of how we do analysis. Previously, in bulk RNA sequencing data, let's say you'd have 100 samples. And even 100 samples is already kind of, first of all, a lot of work to profile experimentally, and it's also kind of expensive.
Manual intervention,
Right, but you're in a situation where you have only 100 columns and 20,000 rows.
Mhm.
And so you can't really do much deep learning across 100 columns, right?
Yeah,
So when previously what people would do when they applied deep learning to this analysis, they would ask questions across rows. So they kind of ask, for example, what is the sequence, the genomic sequence in front of this class of genes versus another class of genes? And neural networks are actually kind of been used in that aspect a lot in the beginning for something called motif analysis. So you could ask questions about different genes with deep learning, but you couldn't really ask questions about different samples. So you couldn't really do the analysis across the samples. But with the advent of single-cell RNA sequencing data, now you have huge matrices or very wide matrices, right, where you have a lot of columns. So this is kind of the first time in molecular biology that you have more data points, more samples than genes. And when you're thinking about it from an analysis perspective, again, you're not in that small n large p problem anymore, and you can kind of use deep learning methods sort of out of the box on this type of data. And so I was involved in projects, and we have kind of made some publications on the topic of applying deep learning algorithms to specifically single-cell RNA sequencing data sets. And I think all of us kind of data enthusiasts are really excited to get our hands on single-cell RNA sequencing data, because now we can actually try to apply complicated machine learning, deep learning models that previously we just didn't have the power to, or if we applied them, we could only ask questions about genes and not really across samples.
Yeah, I mean, that's really interesting. It really like kind of coming to the main point here, that how this advent of deep learning technology supported by the data, the gene expression data, literally change the kind of the whole implementation of AI and machine learning in the research. Because I guess a few decades back it was not possible, because first of all we don't have an easy access to technology that can maybe generate the data very fast and rapidly, and also we don't have a tool or some sort of technology similar to deep learning or neural network that can literally just take the data points and try to profiling the gene expression accordingly. And just follow-up question. When you're talking about this profiling gene expression, just for my viewers who are not in that scientific research field, is it profiling is more of like a prediction of the gene patterns in the sense, in the data science sense, or do you think profiling is something else other than just prediction of the gene pattern?
So yeah, right. When I say profiling, that's actually just a measurement of genes. So that's coming from the technology, that's coming sort of from the machine. It's just measuring, what's the expression of this gene in that cell, and that's what I mean with profiling. What you do with it, whatever the application is of that, what you want to kind of extract from that data, that's a different question. And so, just to give an example, in one of our previous projects, we implemented a autoencoder. An autoencoder is a neural network that tries to kind of learn itself, right? But it has a important constraint that in the hidden layer, you're using a lot lower dimensionality than your input data. Yeah. And so what it does, it forces the network to learn a very efficient compression of the data. And so we applied this to single-cell RNA sequencing data. And then we try to look at, okay, what is the compression that we actually learned from the data? We were really curious, okay, what did the network learn? And what it learned is sort of the data manifold of the data. So it kind of learns different clusters of cells. So for example, if you had a sample that consisted of different cell types, you would actually identify different cell types in that hidden space. And so that was one of the first applications of deep learning to sort of single-cell RNA sequencing data, and it showed really nicely that the algorithm is just optimizing a loss function, right? But the information and the insight that you gain is really biological, right? Because the clusters represented different cell types. So when we the first time we looked into that hidden layer, and we saw that these cells were colored by different cell types, and they were kind of mapping to different locations, we were very excited because it showed that our algorithm was able to extract real biology.
That's great to know. And also, Lukas, as you mentioned that you actually have a research paper associated with that. Just for my viewers reference, so will you be able to like share the research paper link with us so I can actually mention that in the video caption?
Sure, I will send you the link to those papers. No problem.
Thank you. Yeah. So viewers, like please check out the video caption where I would share Lukas' research paper link. Hopefully, you will get more information out of that. And as you like just wrap it up, as you mentioned that like how deep learning is being very crucial and very implementational in your scientific research. And if I think about some other aspect of deep learning, we saw that how DeepMind is basically using deep learning to like do a protein discovery. And I feel like like your research, it's not similar to that, but we are actually going towards the same direction where we're using a technology to understand the hidden pattern of molecular biology, or is it protein structure, or trying to profiling gene expression. So I feel like as a scientific community, I guess we are all going towards that direction where we can easily benefit from from the advancement of deep learning. And it's not possible without the kind of tools we have. I don't know what kind of tools you use. I just wanted to, out of curiosity, either your team is heavily involved in building the deep learning model using PyTorch or TensorFlow, or do you just use regular R and Python programming language to develop your own model from statistical standpoint?
Yeah, I would say that really depends on personal preference. Some people they prefer Python, others they use R. They use maybe different. What we commonly use is Keras, so it's a TensorFlow based. Originally Python implementation, but there's also like a R wrapper around it. That's our go-to, let's say, language for building neural nets. But there's more to the analysis than just building the neural net, right? You also want to visualize the data and analyze sort of the outputs and so on. So I think there it's kind of free to personal preference what people use, at least in our group. And so it's a healthy mix, I would say, between R and Python enthusiasts.
Yeah, I mean, that's a great point, and again, for my viewers, it's not like we should be like a bogged down with one technology. I mean, everybody has like, should have preference to use their own technology, as long as our main goal would be like, consistent, and we can use any sort of toolkits. And there are a lot of different programming languages that are coming out. So many Python libraries we could use to build any sort of deep learning models. But it just, as long as, as Lukas mentioned, as long as we keep our data matrix consistent, it should be compatible with our model, then, you can either use Keras or PyTorch or even TensorFlow. It won't make a significant impact to your model performance based on your technology or language preference. So I think that's all. That brings me to an end of this coffee chat session. Thanks, Lukas, and thanks for providing a lot of information about your academic research. It's a very informative and engaging discussion. I do appreciate your time and consideration for this. And for my viewers, please take a look at the video caption. I'll share Lukas' research paper over there. And if you do have any follow-up questions, please feel free to share in the comment section. And also, if you would like to follow Lukas, he's available. He's actually has an account on LinkedIn. So I would also share his LinkedIn account over there. If you're interested to know more about his academic research, please feel free to get in touch with him on LinkedIn. Okay, that's all for now. Happy Easter again to all of you, and I'll see you in the next coffee chat session. Till then, goodbye. Thanks, Lukas.
Thanks, Sam. Bye-bye.